So every cell in your body has DNA, and the DNA is what you've inherited from your mum and your dad, and it's exactly the same in every cell in your body. But of course, every cell is different, like a nerve cell is different from a skin cell, is different from a blood cell. And the thing that makes it different is the layer of information on top of the DNA, which is your epigenetics, which controls which genes are turned on and off, and that changes what type of cell you have and how it functions. And so in cancer, your epigenetics goes wrong, and that's what can contribute to the cancer occurring. My name is Ruth Pidsley. I'm a group leader here at the Garvan Institute, and I lead a group called the DNA Methylation Biomarkers Group, and we primarily focus on prostate cancer. I work on prostate cancer, which is one of the most common causes of cancer in men in Australia. Cancer is a disease which is caused by changes to your DNA. And I don't just look at the DNA, I look at marks on top of the DNA and how they get changed in cancer. And so we're trying to understand and learn more about these as potential avenues for treatment and diagnostics. The problem I'm trying to solve is that at the moment, the clinical tests for prostate cancer lack diagnostic accuracy. So we're trying to improve the methods for diagnosis and prognosis so that men can be directed to the right treatment at the right time. We've only just started this research project, but already we're starting to use a new technique called digital droplet PCR. You might have heard about PCR from COVID, and it's a way of amplifying small amounts of DNA. And then we're also using another new technology called spatial transcriptomics, which is a very new technique where you can look at segments of tissue, and on those tissues, you can look at every single cell that's there, which of the 20,000 different genes each of those cells is expressing. It's beautiful data. We're going to learn a lot from it. We couldn't do any research without the funds. We need it to help pay people's salaries and to do the experiments that we have planned. I work in genomics, which is a field that's seen a lot of advancements in the last couple of decades, going from looking at single genes to whole genomes, like billions of bases of DNA. So a typical experiment would cost tens of thousands of dollars to do, even on small sample sizes. So the project funded by Rallies is 3 years, but I'm really hopeful that with this money and the results that we can generate from this project, we'll be able to apply for new funding and keep the project going long term. You can't really tackle something unless you understand what you're dealing with. So I think it's critical to understand as much as you can about the disease so that you can find its weak points. For example, in my case, I'm studying the epigenetics of prostate cancer, and recently there's been the development of epigenetic drugs that can reprogram your epigenetics, and that's something I'm really excited about, and it wouldn't have been possible if people hadn't done that basic research in trying to understand the disease. I think we've seen huge progress in cancer research and treatment over the last few decades, and I think that's only continuing, especially with the development of epigenetic drugs and the increased understanding of the role of the immune system in cancer. So I'd like to say a big thank you for everyone who donates money towards Box Rallies and our funding. It's really critical, and we couldn't do this work without you.